Cell theory claims that all cells come from pre-existing cells, and therefore new cells must be created by the division of a pre-existing cell, the process called cell division. And it's obvious in growing organisms that cell division is necessary for the elaboration of tissue, the increase in biomass. However, even organisms that have stopped growing need to replace cells that die every day by new cells through the process of cell division. The cell cycle refers to a sequence of events that occur between the time a new cell is created from cell division until the time that that newly created cell divides again. And most of the cell cycle is the process of interphase. Interphase simply refers to the time period where cells aren't in the process of dividing and mostly carrying out their normal functions. Now, these divisions within interphase are as follows. There are G1 and G2 phases of interphase, as you can see here. Those are normal cell functions, the cells carrying out their normal everyday activities, but also preparing for things like cell division and DNA replication by doubling organelles and preparing the production of proteins. Now, another G phase that you see here in the diagram is a G sub 0. And this is for cells that do not undergo regular cell division. They simply stay in that G1 phase for an extended period of time. The S phase of the cell cycle refers to a synthesis phase, and this is the period of time when DNA is being copied through the process of DNA replication, again in preparation for cell division. And finally, the M phase of the cell cycle refers to mitosis or meiosis, the forms of cell division where the cells actually divide. Now, we need to be clear on our definitions of just what a chromosome is. Chromosomes, the term refers to DNA packaged in protein, and there is quite a hierarchy of structures to consider. First is the DNA double helix, a simple sequence of nucleotides arranged in that double-stranded helix. However, this DNA inside the nucleus is not just the bare molecule, but it coils around these proteins called histones. Now, there are many histones around which the DNA coils into this beads on a string model. And then those beads further coil into a very thick strand, and then that strand of coiled beads further begins to coil, and then more proteins are involved, shown here as the green band. More proteins are involved that further scaffold the DNA around it and tighten it into a form of chromosomes called chromatin. A chromatin is the loose form of our chromosomes in the nucleus during the interphase. Again, remember most of the cell cycle is an interphase when DNA is carrying out its normal function. The chromatin in the nucleus can be seen here in more dense and more loose regions, but all of this is chromatin, the loose form of our chromosomes. Now, chromatin will condense further into chromatids in order to prepare for cell division, as you can see here. Those chromatin strands will further wrap around even more protein structure into a highly condensed form of chromosomes called chromatids. And what we see here in this characteristic four-arm or X-shaped structure is the appearance of two sister chromatids bound together. Those two sister chromatids are actually the two copies of DNA formed during DNA replication. So both identical copies of a single DNA molecule are bound together as sister chromatids. Now, those sister chromatids are joined by a constricted region called a centromere that you can see in this diagram here. And so we see the two sister chromatids bound together at this point of constriction in the middle called the centromere. And that's often represented in a cartoon fashion as this kind of characteristic X shape with just simply a dot in the middle representing the centromere. In this case, this cartoon is actually representing a single chromosome we call this a single chromosome consisting of two sister chromatids. And then as you can see, here are the 46 human chromosomes arranged in 23 pairs of chromosomes, and you can see they're in this highly condensed chromatid form and thus ready for cell division.